The human genome was first sequenced in 2003 after 8 years of active sequencing at a cost of $1 billion. After the completion of the Human Genome Project, the cost and labor associated with sequencing new genomes substantially decreased, and as a result, thousands of other genomes have been sequenced in the past 2 decades. At the same time, many other next-generation sequencing technologies have been developed to learn even more about genomes, such as finding out which parts of DNA are active, how different parts of DNA interact with each other, and how DNA is structured in 3D space. All of these efforts have generated massive amounts of data, but despite that, we still don't know exactly how the information encoded in DNA gives rise to the incredible diversity of life on Earth. That's where I come in. My name is Amanda Kowalczyk. I'm a fifth-year PhD student studying computational biology, and my work takes advantage of the enormous amount of genomic data that already exists to figure out how our genomes work. My goal is to create state-of-the-art methods to turn a mountain of data into answers to biological questions. I take a comparative approach to studying genomic data. In other words, I compare the genomes of numerous species to look for similarities and differences that could explain genomic function. For example, in my most recent project, I studied mammalian hairlessness. Some mammals, like whales, naked mole rats, rhinos, and elephants, have much less hair than others. And prior to my work, we had no idea what in these species' genomes was causing them to be hairless. To find out, I used sequenced genomes from 62 mammal species, including 10 hairless mammals, to develop statistical methods to identify which parts of the genome are different in hairless mammals compared to hairy mammals. I was able to identify many genes that we know are hair-related, such as keratins and hair follicle proteins, and for the first time link them to natural mammalian hairlessness. Since I developed computational methods, the same code I wrote to study hairlessness can also be used to study any characteristic I'm interested in. I used my methods to study extreme lifespan in mammals and found a myriad of cancer control mechanisms essential to long life. I've contributed to projects to identify the genetic basis of vision by comparing genomes of blind subterranean mammals to sighted mammals. I have collaborators who use my methods to study traits ranging from sleep rhythms to vocal learning to survival at extreme altitudes. All of this is thanks to the power of huge amounts of data and rigorous methods to analyze them. The scientific community has come a long way since first sequencing the human genome, but we still have a long way to go to fully decipher the genetic code. I hope that my methods are one meaningful step toward that goal.